Hello, my name is Simon Sharples, and I'm a postdoctoral fellow in the Miles Lab at the University of St. Andrews. And I'm pleased to share with you work that was recently published in eLife that describes how active properties shape the early recruitment of spinal motor neurons in the mouse spinal cord. As I mentioned, I'm speaking to you from St. Andrews, which is a wee town on the east coast of Scotland that is particularly well known for its golf. And in golf, there's a saying that goes, drive for show, putt for dough. And this is because the best golfers aren't the ones that can smack the ball down the fairway. Take Happy Gilmore, for example. The game is in fact won by those that have mastered the short-range shots near the green, which require much more precision and finesse. However, the best players are the ones that can do both, and I think this speaks to the importance of our ability to modulate the balance between precision and vigor of our movements. But for those studying motor control, it is well accepted that there are two primary mechanisms encoded at the level of the motor unit to modulate the amount of muscle force that we produce. The first is known as rate coding, with higher motor unit firing rates translating to greater force, muscle force, or power. The second, which I will focus on in my talk, is through the recruitment of additional motor units. The traditional view, initially described by Henneman in the late 50s, suggests that motor unit recruitment follows a size-based recruitment scheme, with motor units composed of smaller motor neurons recruited before those of motor units with larger motor neurons, essentially following Ohm's law. Put simply, smaller motor neurons have higher input resistance, such that smaller currents can produce larger changes in membrane potential. Subsequently, Burke and colleagues demonstrated that there are several motor unit classes that can be identified based on muscle twitch properties, which also follow an orderly recruitment scheme, with motor units of increasing force generating capacity and twitch speeds recruited in an orderly fashion. However, in contrast to what would be expected from a purely size-based recruitment rule, motor neuron size actually turns out to be a poor predictor of recruitment threshold of functionally defined motor unit subtypes. But there are some assumptions that were made at the time these recruitment schemes were first described that may not be entirely accurate. The first is that motor neurons are not passive integrators. As we know quite well now, motor neurons are endowed with active properties that not only amplify synaptic input, for example, persistent inward currents, but there's also others that can diminish their influence. The second assumption that was made is that fast motor neurons are simply scaled up versions of slow motor neurons, but we know that this is far from true, as motor neurons that innervate different muscle types express different complements of voltage-sensitive ion channels. For example, fast and slow motor neurons express different complements of ion channels that have been suggested to underlie key differences in intrinsic properties, such as the medium after hyperpolarization. We therefore asked what properties and underlying ion channels differentiate motor neuron subtypes and shape their recruitment. To address this question, we performed whole cell patch clamp recordings from lumbar motor neurons of postnatal mice. And in these preparations, we can identify two motor neuron subtypes based on the latency to repetitive firing when injecting a 5-second depolarizing current step. We used an approach established by Leroy and colleagues to identify fast motor neuron subtypes based on a delayed onset for repetitive firing and accelerating firing rate, and slow motor neurons can be identified based on an immediate onset for repetitive firing and a relatively stable or even adapting firing rate. And for the duration of this talk, I will refer to them as fast and slow type, with fast depicted in purple and slow in green. We studied the maturation of intrinsic properties of these two motor neuron subtypes across the first 3 weeks of postnatal development, which is not only a period of time that we can obtain viable recordings from motor neurons in slices in vitro, but is also a convenient model to study adaptive processes that may contribute to the generation of complex movement that would be expected to rely on the ability to appropriately recruit motor units. We therefore hypothesized that key properties that shape motor neuron recruitment would emerge in parallel to complex motor behaviors. To assess recruitment, we use slow depolarizing current ramps, which allow us to directly measure how much current is required to elicit repetitive firing and is known as the rheobase. This approach also provides a continuous readout of the subthreshold voltage trajectory leading up to the recruitment of firing, and allows us to detect any nonlinearities that might be produced by voltage-sensitive ion channels that could influence their recruitment. We also measured the whole cell capacitance for all cell studies, which provides a measure of motoneuron size. So, what did we find? Well, during the first postnatal week, in line with what would be expected based on the size principle, motoneuron rheobase is correlated with cell size measured by whole cell capacitance. However, while we could identify both of the subtypes based on delayed and immediate firing profiles, there is no difference in the rheobase or capacitance between the two motoneuron subtypes at this stage. I'd also like to highlight that we did a fairly comprehensive analysis of intrinsic properties in this study. Consistent with what we see with the rheobase and capacitance, globally, these two subtypes are functionally homogeneous at this stage. In the second week, we start to see the emergence of a more orderly based recruitment scheme, with the slow type at the lower end, here on the left, and the fast type on the upper. The staggering of this relationship here is due to an increase in the rheobase of fast motor neurons only, and this increase in rheobase coincides with an increase in whole cell capacitance. Things continue to change into the third week. While the overall orderly recruitment scheme is preserved, the strength of this relationship begins to degrade. While there is an increase in rheobase in the fast types progressing into this third week, there is not a parallel change in motor neuron size indicated by whole cell capacitance. So if not size, what can account for this increase in rheobase of the fast motor neurons between weeks 2 and 3? Well, as I mentioned before, the subthreshold membrane potential trajectory may provide some insight into this. In fact, previous work from the Heckman lab and also the Meehan lab have described a role for persistent sodium currents in producing this acceleration of the membrane potential approaching the onset of repetitive firing. So maybe changes in persistent inward current could account for changes in rheobase during development. To address this possibility, we assess persistent inward currents in voltage clamp using an established approach whereby motor neurons are subjected to a triangular voltage ramp starting from -90 millivolts, moving up to -10 millivolts. So on the left here, we have a raw current trace from a fast motor neuron purple and a slow motor neuron in green. And on the right here is a leak subtracted trace with the negative deflection produced by a persistent inward current. We measured a number of different features of the PIC here. What we found was that persistent recurrence in fast motor neurons were activated at a much more depolarized voltage compared to the slow types, which we hypothesized might contribute to the higher recruitment threshold. Now to test this, we would need a way to block or manipulate persistent inward currents. However, PICs are produced by a number of voltage-sensitive ion channels. We chose to assess the contribution of two types of channels that are key contributors to the persistent inward current. The contribution of NaV 1.6 channels was assessed using a blocker called 4,9-hydrotetrodotoxin, and the contributions of L-type calcium channels with a blocker called nifedipine. So here on the left, we have our leak subtracted current from our voltage clamp recordings. In red is the current measured at baseline, and in black, the currents measured after blocking either sodium or calcium channels. What we found was that although both channel blockers reduced the amplitude of the PIC, particularly in fast motor neurons, blocking NaV 1.6 channels caused depolarization in the PIC onset voltage in the fast type only. And in line with their measures of PIC, we find that blocking these NaV 1.6 channels led to a significant increase in the rheobase of fast type motor neurons, suggesting that these channels play a key role in shaping their recruitment by modulating the onset of the PIC. We next asked if changes in PIC onset voltage could account for their increase in rheobase at week 3. However, in contrast to what we would have hoped for, we did not find any changes in PIC onset voltage in either motor neuron subtype between weeks 2 and 3, although differences between subtypes are preserved at both time points. Okay, so if it's not size, and it's not the PIC, what could possibly be leading to this increase in rheobase of fast motor neurons between the second and third postnatal week? Well, it could be a number of things, but to make a long story short here, what we noticed when assessing the input resistance of these fast motor neurons at week 3 was that they had this prominent sag potential when injecting hyperpolarizing current steps. Now, these sag potentials are produced by these hyperpolarization-activated inward currents, otherwise known as IH. and are typically activated at very hyperpolarized potentials, usually below negative 80 millivolts. However, what you can see in this example here on the left is that we see a sag potential when we are bringing the membrane potential down from negative 60 to this particular cell's resting membrane potential. Importantly, this sag potential can be eliminated by a drug that blocks the ion channels that produce the underlying IH. And what you can see here in this graph is that this sag potential and the drug's effect is specific to the fast motor neurons, which are highlighted in purple. So this led us to ask whether an increase in this H current could account for changes in rheobase between weeks 2 and 3. We therefore measured IH and voltage clamp using a series of hyperpolarizing voltage steps in both fast and slow motor neurons at weeks 2 and 3. And the H current can be measured in a very similar way in voltage clamp as it is in current clamp, where the cell is subjected to a series of hyperpolarizing voltage steps, and H current measured as the negative deflection in current from the start to the end of the step. And what you can see in this graph on the right is that the amount of H current measured when stepping the membrane potential from negative 60 millivolts to negative 70 millivolts, which is close to the resting potential in these motor neurons, is that we have a significant increase in how much H current there is in the fast motor neurons at week 3 only. Okay, great. So now we have identified a current that increases in fast motor neurons only between weeks 2 and 3. But can it influence rheobase? And can that possibly account for the increase in rheobase at week 3? We next flipped back into current clamp and used our depolarizing current ramps to measure rheobase both before and after application of the IH blocker, and found that blockade of IH led to a significant decrease in the rheobase of fast motor neurons, but not slow motor neurons during week 3, such that rheobase was no longer statistically different from slow motor neurons at their respective baseline. So what we think this means is that because the H currents are on at resting potential, they may just act as some sort of a shunt that might diminish the effectiveness of inputs to change the membrane potential, and as a result, delay their recruitment. Okay, so to summarize, what I've shown you today is that the recruitment of motor neuron types is multifaceted, and not only dependent on passive properties, but also active properties. And this supports the notion proposed back in the 80s that fast motor neurons are not simply scaled up versions of slow motor neurons. And finally, orderly recruitment of motor neuron subtypes is established during the first 3 postnatal weeks in the mouse. And this is due to an increase in the size of fast motor neurons during the 2nd week, and the differential integration of persistent inward current and IH amongst subtypes. Before I finish, I would like to acknowledge support from my lab, in particular, my supervisor, Gareth Miles, in addition to my sources of funding. Finally, if you'd like to learn more about the results of the project that I have shared with you today, you can check out the full paper published in eLife. Or, if you'd like to learn more about my research in general, feel free to check out my research website or follow my updates on Twitter.